Hi class, in this discussion we are going to be talking about proteins. As we look at proteins, just for a bias alert, they're my favorite macromolecule. I think they're fantastic. When I was an undergraduate, I studied biochemistry. That was one of my majors. And I took a lot of classes on protein structure. So as we look at proteins, they are a polymer of amino acids. Poly meaning many, mer meaning subunits. So proteins are made of many amino acids being combined together. Every once in a while, we'll call a protein a polypeptide. Referring to the special kind of covalent bond that holds them together. A peptide bond holds the amino acids together. It's just a kind of covalent bond. Now, as we look at amino acids, all amino acids are going to have the same generic structure. There'll be an amino group. So here in green, I'm circling the amino group of two separate amino acids. And then we have a carboxylic acid functional group that I'm circling in red with our amino acids, and then in blue, I'm going to circle the R group. The R group is just a generic term that refers to the variable part of the amino acid, the part that changes from individual amino acid to individual amino acid. So as we look at these amino acids, they all have a central carbon atom with three different things attached to them: an amino group, a carboxylic acid group, and that R group. And the properties of individual amino acids are going to be determined by the R group that varies from amino acid type to amino acid type. As we look at proteins in our bodies, proteins, and actually I would be remiss if I didn't mention this. I'm going to go back a slide here. So as we look at amino acids, we need to emphasize that in the human body, we have 20 amino acids that are used to make the proteins in our bodies. Now, that's not to say that there are not more than 20 amino acids in existence, but within human beings, we use 20 different amino acids to make the proteins of our bodies. All right, let's move on. So as we look at proteins, proteins are going to have some structure. Their structure, or the shape, is referred to as conformation. And when we think of this conformation, not conformation, the conformation of a protein is going to be its shape. Structure determines function. The shape determines a molecule, or a structure determines what you do with that molecule or structure. So when we think of the conformation of a protein, it's crucial to its function. Proteins can reversibly change shape. They can oscillate their shape just a little bit. There's a little bit of atomic motion, or the quantum jitters as it's sometimes referred to. And then depending on chemical environment or electrical environment, we can open or close, or we can alter protein shapes more dramatically. This is commonly done, for example, when we have membrane channels in our bodies open and close within our cell membranes based on electrical activity across the cell membrane. If we denature a protein, we are going to cause an extreme change in its shape, or an extreme conformational change, so much so that we've destroyed the protein. So when we think of denaturation, I want you to think of a protein that's had its shape altered so much that it no longer performs the correct function. Typically, we denature proteins with heat or pH. This is a classic reason why we cook our foods. By cooking food, we denature the proteins in the food. Not only does it make it easier to chemically digest the food, but it also is going to denature the proteins that keep the microorganisms in the food alive. So by applying high heat, we change the shape of the proteins, and then by changing the shapes of the bacterial and microbial proteins, we kill those microbes that are in our food. This is one of the, again, why it's really good to cook our food. When we cook an egg, and this is another classic example, we have albumin molecules that are a protein. And these albumin proteins are going to be dissolved, or suspended, I should say, in aqueous solution. And as we heat up the albumin protein, they start to change shape. And eventually, they become, for lack of a better analogy, kind of spaghetti noodles that then connect to each other. And as they start to connect to each other, they go from small proteins to a much larger mass of protein. When they're small proteins, they're water soluble. When they're the larger mass, they are insoluble and fall out of solution, and you can see them as a white product within the egg albumin, or the egg white, I should say. So we have natured and denatured albumin as we cook an egg. Now, as we look at a protein, all proteins are going to have some structural stages they go through. As the protein is being manufactured at a ribosome, the ribosome will plug individual amino acids, represented by these beads on a string right now, into the protein. And these beads on a string determine the primary structure of the protein, just the sequence of amino acids, the order that they're connected to each other in. And then this long string of amino acids can form some secondary shapes. They can coil on themselves, and if it coils on itself, it's referred to as an alpha helix. Or it can zigzag back and forth on itself. And if it zigzags back and forth on itself, it's referred to as a beta helix. I'm a little bit crustier, so I was taught beta pleated sheet when I was an undergraduate. And those are the two most common secondary structures made from the primary structure of a protein. And if you take those secondary structures, you can take an alpha helix and a beta pleated sheet and combine them together, and you can have multiple alpha helices and multiple beta pleated sheets and combine them all together. We still are going to have one continuous strand of amino acids, though. And that's a key point. We still have a continuous strand of amino acids. But as we look at these alpha helices and beta pleated sheets, they can interact with each other based on hydrophobic-hydrophilic interactions. The hydrophobic-hydrophilic interactions are determined by the R group, or the side chain group of the individual amino acids that were put into the primary structure of the protein. And some proteins just stop at the tertiary structure, but other proteins are going to take multiple links of beads on a string, or multiple polypeptides, and combine them together to make a quaternary structure. So when we think of a quaternary structure, I want you to think of two or more polypeptide chains bound together by ionic bonds, or those hydrophobic-hydrophilic interactions. The classic example of a quaternary protein is going to be hemoglobin. So here's a hemoglobin molecule. It's made of 2 beta subunits and 2 alpha subunits. And these quaternary structures are not as common as tertiary structures. All proteins have a tertiary structure. Not all proteins have a quaternary structure. Now, as we look at protein structure, the structure of a protein helps determine some of its chemical properties. Or, excuse me, when we think of protein functions, the structure of our body is determined by proteins. Proteins are the most common macromolecule in our body. Some common examples include both keratin and collagen, 2 common structural proteins. So as we look at keratin, we use it to make our hair, our finger and skin, our fingernails and our toenails, and we also use it to help toughen up the surface of our skin. Collagen is a very common protein in our bodies. It has in the deeper layers of the skin, bones, cartilage, teeth. It helps to bind all the different tissue types of our bodies together. And we have multiple forms of collagen within our bodies. We also use proteins for communication in our bodies. So we have protein-based hormones in our bodies that travel through the bloodstream to send information. We can use proteins for cell-to-cell information or cell-to-cell signaling. We also have receptors on the surface of cells, which are made of protein molecules. As we look at membranes, the cell membrane in our bodies, we use channel proteins, proteins within the cell membrane that help to determine what does and does not go in and out of the cell. We also can use proteins to carry specific chemical compounds across the cell membrane, or even within the bloodstream itself. And then finally, we can use proteins as catalysts. As we think of these catalysts, proteins that speed up chemical reactions, or enzyme-based enzymes that are made out of proteins, catalysts that are made out of proteins can be referred to as enzymes. Sorry about that. I had a brain hiccup right there. Enzymes are protein-based catalysts. So proteins have a lot of functions. In addition, proteins can be used for recognition and protection. So as we think of these glycoproteins, a glycogen or a carbohydrate attached to a protein can help identify cells for our immune system so that our immune system knows what does and does not belong in our bodies. All the antibodies in our bodies are also going to be made out of proteins. We use proteins for movements. We have actin and myosin within our skeletal muscle and cardiac muscle tissues that we use to contract our muscles. We also are going to have dynein and kinesin that are used as motor proteins that move transport vesicles from one part of a cell to another part of a cell. And we also can use motor proteins to change the shape of our cells. Think of white blood cells that are sending out pseudopods to phagocytize or swallow a bacterium. We use motor proteins to help change the shape of those cells. We're also going to have cell adhesion. We can use proteins to bind separate cells together. For example, a sperm can be attached to an egg via some proteins on the cell membrane surface. And we use proteins to keep our tissues from falling apart. For example, we can have collagen, as I've already mentioned, and there's a wide variety of other proteins within our body that help to anchor and link the cells of our bodies together. We'll talk about them more when we get to organelles and parts of the cell. And that's all we have for this recording. If you have any questions or comments, please feel free to post them on the class discussion board or shoot me an email. And as always, happy studies.